Hello everyone and welcome to the video which is an overview of glycolysis. Glycolysis is partial breakdown of one molecule of glucose into two molecules of pyruvic acid. A simple way to remember glycolysis is by looking at its literal meaning. Glyco means sugar and lysis means split. Glycolysis is the first of three steps in a process known as cellular respiration. The products of glycolysis will be used in the second step of cellular respiration called the Krebs cycle, which will lead on to the final step called the electron transport chain. Both Krebs cycle and the electron transport chain occur in the mitochondria of the cell. Essentially, glycolysis is an energy conversion pathway that is common in both prokaryotes and eukaryotes. In eukaryotes, this process occurs in the cytosol of the cell, shown below in the diagram. Let's have a look at an overview again before we get into the pathway. Glucose, which is a 6-carbon molecule, is broken down into two 3-carbon molecules called pyruvate. Along with two molecules of pyruvate, two molecules of ATP and two molecules of NADH are yielded.
Glucose enters the cell through specific transport proteins and has one principal fate. It is phosphorylated by ATP to form glucose 6-phosphate. A phosphoryl group from ATP is transferred to the hydroxyl group on carbon 6 of glucose. Next, the enzyme phosphoglucose isomerase acts upon glucose 6-phosphate to form fructose 6-phosphate. Isomerases are enzymes that rearrange the structure of the substrate without changing the molecular formula. The 6-ring glucose 6-phosphate is rearranged to a 5-ring fructose 6-phosphate. The next step is going from fructose 6-phosphate to fructose 1,6-bisphosphate. In this reaction, ATP is converted to ADP and is facilitated through an enzyme called phosphofructokinase. In this step, a phosphate group is added to one of the free carbon molecules in the ring. Following this, the fructose 1,6-bisphosphate enzyme is split by the enzyme fructose bisphosphate aldolase. The enzyme splits carbon-to-carbon bonds in fructose 1,6-bisphosphate, forming two products, namely dihydroxyacetone and glyceraldehyde 3-phosphate. The next reaction involves converting dihydroxyacetone to glyceraldehyde 3-phosphate. This is conducted by another isomerase, the triose phosphate isomerase. The next enzyme we meet is the enzyme glyceraldehyde 3-phosphate dehydrogenase. In this reaction, a hydride ion is moved onto the electron receptor NAD+, creating 2 NADH. 2 NADH is created because one glucose molecule creates 2 glyceraldehyde 3-phosphate, one from the aldolase reaction and one from the triose phosphate isomerase reaction. Inorganic phosphate provides the phosphate group, which replaces the hydride group on the molecule. Energy for this to occur is provided by the reduction of NAD+ to NADH, giving us the molecule 1,3-bisphosphate glycerate. In the next reaction, the enzyme phosphoglycerate takes away a phosphate group from 1,3-bisphosphoglycerate to form 3-phosphoglycerate. In this process, ADP is converted to ATP. This is the first time ATP is created. The enzyme phosphoglucomutase is essentially an isomerase, thus it creates the isomer 2-phosphoglycerate from 3-phosphoglycerate. A hydroxyl group is removed from 2-phosphoglycerate by the enzyme enolase through the production of a double bond, creating phosphophenol pyruvate. This is a dehydration reaction, thus H2O is removed from 2-phosphophenol glycerate. The last reaction of glycolysis, the enzyme pyruvate kinase removes phosphate from the phosphophenol pyruvate and adds them to an ADP, forming ATP. 2 ATPs are created because phosphophenol pyruvate will be created for every glucose molecule. This leaves us with the end products of 2 pyruvate molecules. Since there are 2 molecules of glyceraldehyde 3-phosphate, this reaction will occur twice per glucose molecule. Pyruvate is oxidized to acetyl coenzyme A, which enters the citric acid cycle for further oxidation to CO2.
To conclude, glycolysis is an anaerobic process that does not involve oxygen. In the presence of oxygen, pyruvate can be further oxidized to CO2. In the absence of oxygen, pyruvate can be fermented into lactic or ethanol. This video was created by 3 4th year biopharmaceutical students from DKT, and we hope this will help.